Okay, so I think we can start. I see already a lot of people connected for this HITRI seminars. As you know, the HITRI seminars are organized by Manjit, that I hope is still online. I don't see you anymore.
I'm here.
Oh yeah, they are there. You are there. So Manjit asked me to give a short introduction to this seminar that is within the HITRI project. For those, maybe some of you are not familiar with the HITRI project. This is a European project, belongs to the class of the infrastructure projects. And it's very interesting because it involves all the European centers doing carbon ion therapy, which means Heidelberg, Marburg, Medostron, and CNAO. Actually, CNAO, Sandro Rossi is the PI of the project, and also other research centers connected, including GSI, where both myself and Insa belong. One of the main goals of the infrastructure programs in the European Union is to allow translational access, which means that both researchers and medical doctors are able to use the facilities involved in the project and can use either research beam time or what we call clinical beam time, which involves also training and, let's say, improvement of the knowledge of the heavy ion therapy in Europe. At the end of the seminar, I think Insa will show a few slides about the translational access, in particular in GSI, but of course her seminar will be about the organoids. So first, just a few words about Insa. I mean, she works in our department at GSI, and she's a group leader. She leads the group of stem cells. And even though she got the PhD in food chemistry, she essentially always worked on stem cells. First at NIH in Bethesda, and then in Germany, in particularly in Leipzig. And she worked on embryonic stem cells and on induced pluripotent stem cells, the so-called iPS. I think all of you are familiar with the difference between embryonic stem cells and iPS because it's kind of a well-known story. But then she moved to GSI, where she's now group leader, and she's completely focusing her work on organoids. Organoids are a completely new biological system, which is based on human stem cells. So it's a human model. So in this sense, for some aspects, it can be considered superior to the animal models, because instead of being an animal, now it's really human. And of course, it's much superior to the cell systems that are not even, that cannot really resemble a human organ. They are extensively used in different branches of medicine, and Insa will show some example, and also in toxicology, but they are not really very much used as yet in radiation research, especially in preclinical radiobiology for particle therapy, which is now the main focus of this lecture and of the work of Insa. She will focus on the cerebral organoids, so the so-called mini brains. And she will show how these mini human brains, much smaller than an average human brain. Maybe some of you know some people that have very small brains, but that's even smaller. And she will show that they can be useful both for studying the NTCP, let's say the toxicity of the treatment, and for studying the TCP, so the capability of using the beams for tumor control. So I think is a very innovative system where Insa is a world leader, we just got, together with MD Anderson, an NIH grant for studying human organoids in particle therapy. And this was one of the very few grants that NIH gave in the field of heavy ion therapy, and one of the few times that a European group receives funding directly from NIH. So this is to show how this system is appreciated. And Insa will give us an idea of how it works. So I'm sorry I was a bit long, but without any further ado, Insa, please.
Thank you so much, Marco, for this nice introduction. I'm very happy to introduce the human organoids to you, and I hope I can convince you that it would be really not a wise decision to not use them in radioprotection studies and in studies regarding particle therapy. But I will start out with a very simple question. And I hope I can, oh, now it's frozen. No, here we go. My computer may be a little slow. So I will start with a very slow question. What are organoids? And even though I always tell my students, please do not Wikipedia in a scientific setting, when it comes to the definition of organoids, actually Wikipedia is pretty good, because an organoid, according to Wikipedia, and it is true, is a miniaturized and simplified version of an organ with, and this is a very interesting feature, realistic microanatomy. So an organoid you can derive by using pluripotent stem cells, and I will tell you what this is a little later. Or you can use organ progenitors already. These are usually primary cells from patients with a certain disease. The more scientific view on organoids was published by Lancaster and Knoblich, who are actually pioneers in the organoid research. So they need to fulfill 3 requirements. They have to have multiple organ-specific cell types. They need to be capable of recapitulating at least some of the specific functions of the organ, meaning contraction or endocrine secretion, such as insulin or neural activity or anything like that. And this is really important, the cells need to be grouped together, spatially organized, and we call it, stem cell researchers, we call it the capacity to self-organize and differentiate in a spatially and temporally controlled manner, similar to that you find in vivo organ development. And these organoids come in really different flavors, and I hope you can see my pointer here. So we have actually very simple organoids like the colonoids or the gastroids that are actually only comprise of a single layer of epithelial cells. But we also have quite elaborate and complicated organoids like the brain organoids. And even though we love to use the word mini brains, this may not always be the best to do. So it's cerebral organoids, because the mini brain, it's not a mini brain. Somebody was asking me recently, do these mini brains sense pain, or do they feel something? Of course not, but this is what you get when you call something mini brain. So for the more, as these simple ones are usually made by primary cells, the more complex organoids are usually made by embryonic stem cells or induced pluripotent stem cells. In our lab, we do cardiac clusters or organoids. that already are ready within a short time, 15 days, but also these need to mature a long time. So at day 100, you can really tell that these are real heart cells and heart organoids. Down here, you see the picture of the brain organoids in suspension culture. And you can see here, it takes a long time for them to mature, but brain development also takes a long time. So that's perfectly physiological. As I said, organoids are generated starting sometimes from pluripotent stem cells. We use embryonic stem cells, which are generated from the inner cell mass of a fertilized egg at day 5 to day 7 of human development. This is how they look like in culture. So they are very tiny little cells here. But of course, you can also take somatic cells, fibroblasts, skin cells, whatever there is, and you can reprogram them to make cells that act like embryonic stem cells, so that are pluripotent, and these are human-induced pluripotent stem cells. All of these protocols to generate organoids will go through embryoid body formation, which again resembles the blastocyst. And then with different differentiation cues will form organoids of certain varieties. And of course, the use of human-induced pluripotent stem cells was a great advantage because now you can derive these cells from patients with diseases that have usually a hereditary background, so a genetic background. There is a lot of diseases that you can mimic with cerebral organoids, the ones that we use. For instance, Alzheimer's disease, Parkinson's disease, schizophrenia, or autism spectrum disorder. So the whole thing. And there are other organoids where you have a plethora of diseases that you can mimic in the dish. But these organoids can not only mimic diseases, they are also already used in clinical studies or in the clinics. And one example for this is with patients with cystic fibrosis to test whether certain drugs are working or not. Cystic fibrosis patients have normally mutation in the cystic fibrosis transmembrane conductance regulator. So they are treated with CFTR modulating drugs, but these drugs are really expensive. And Hans Clevers, who did this or who invented this assay years ago, told us that the daily dose of one of these drugs would have the value of 2 iPhones. So you could buy 2 iPhones daily. So of course, this is something that health insurance companies really don't want to cover if it doesn't work. So the simple test that you can do is you can create rectal organoids, or in the meantime already, you can also just use the nasal swab. So if you don't want to do a corona test, but you want to do organoids from your nasal swab, then you end up with these kinds of organoids. And these organoids in a normal, healthy person will swell upon forskolin treatment. In a cystic fibrosis patient, this swelling is not observed. So they don't swell, they can't do this. And so the simple test of responders and non-responders for these drugs is you use this forskolin inducing test. So if you don't use a drug, then nothing will happen. If you use a drug and there's no responder, still nothing will happen. But if the patient does respond to the drug, then you see the swelling. So you can very quickly distinguish the people that will respond and from those that will not respond, and you will save a lot of money. And now, as these organoids are already in the clinics, in practical use, why not also use these organoids in research and in preclinical studies in radiation biology and radiation protection? Because of course, what we now rely on is, yes, we have the impact of radiation. We see in behavioral testings some effect of this. And we also use animal models, of course, to describe the effects. We can see, of course, in functional MRI scans, the region of the brain that is affected. And we can describe something like radiation necrosis or detrimental effects on behavior. But if it comes to really see what the underlying mechanisms are, we're pretty much at loss, because we rely on animal models, and you will know that the brain of a mouse has very little to do with the brain of a human being, or we even rely on 2D cell cultures. So why not take the brain organoid as a new model that bridges these more simplified models and the more complicated in vivo situation, and use this to find out countermeasures that can be implemented then as new mitigants. And you can do that quite nicely with brain organoids, because they really recapitulate the entire in vivo brain development. So you start from a very simple neuroepithelial cell, and by differentiating them, you get all the layers and cells that you would want to see in a brain. So the various types of neurons, the glia cells, oligodendrocytes and astrocytes. And you see that the longer you differentiate these cells and cultivate these cells, you will earn on areal identity and maturation. And this is why we decided to establish a protocol that was originally published by Madeline Lancaster et al. Using human pluripotent stem cells, we use embryonic stem cells. We put them together with microfilaments as a scaffold, and this gives micro-patterned embryoid bodies. And then it takes a long while, but you will get engineered cerebral organoids that are pretty nice and have all the features of brain cells. And here you see a slice of one of these organoids. In pink, you see proliferation zones that are PAX6 positive. PAX6 is a marker for neuronal progenitors. But you will also see that outside of these proliferation zones, you have these patterns of MAP2 positive cells, and this is a marker for mature neurons. So we use this approach to tackle two issues. One is normal tissue protection, and this is a problem here, the radiation necrosis that appears, especially after particle therapy. And this is really detrimental, but because we don't know where it stems from and what are the patients that will suffer from this. And then we want to tackle something that belongs more to the topic of tumor control. That is radiation effects on glioblastomas. And I will give you examples for both of them. But I will start with the radionecrosis, it is a problem, especially in particle therapy. For instance, here, the study by Kralik et al. Identified 31% of the pediatric patients that underwent proton therapy that also then developed a radiation necrosis. And these radiation necrosis start late, so 3 to 11 months after the impact of radiation, also after the radiation treatment. Chemotherapy is a confounding other measure. So chemotherapy is a risk factor as well. In the study by Gunther et al., it was 43% of the examined patients that developed these radiation necrosis with a median of 3.8 months. And the proton beam therapy was associated with a higher likelihood to develop this radiation necrosis, and age mattered. So patients younger than 3 years of age have are more prone to develop radiation necrosis. In the study of Niazi and et al., they tried to link the radiation, the brain necrosis in proton beam therapy with the dose applied. And what they did was they transformed the volumetric dose distribution into an equivalent uniform dose, as shown here. And if you then plot this equivalent uniform dose to the probability to get radiation necrosis, they found that this is a good prognostic parameter for patients either with intracranial or extracranial radiation. The only caveat of the study was that, or one of the caveats, let's put it that way, was that they used a constant RBE value of 1.1. In another study, the same group, Nierko et al., tried to link brain necrosis to an elevated LET and thus an elevated RBE. And here you can see 4 examples from patients, 2 with head and neck cancer and 2 with CNS cases. In red, you see necrotic voxels, and in black, you see the non-necrotic voxels. And as you can see already from these pictures here, they are very heterogeneous. So the patients are, even with the same tumor entity, are very different. And in the end, there could not be seen a correlation between the LET and the tolerable dose at 50%. So the tolerable dose where 50% would get necrosis or would get no necrosis. So they said that, yes, there is not a real prediction or a real correlation between LET and brain necrosis, simply probably because the interpatient heterogeneity may obscure the LET effect. And the reason for this, or one of the reasons that they concluded, were uncertainties in the origin of the necrosis. And this is actually where the brain organoids come in and maybe can help. And that's why we used these engineered cerebral organoids for radiation experiments. And we did X-ray irradiations and carbon irradiations at GSI. And we were lucky and happy to be able to do a lot of proton irradiations at the Heidelberg facility. And here you can see the scheme. We irradiated either organoids in day 20 that resembles more or less the stage where the embryo develops its brain and is very sensitive to radiation. And day 80, which is a more mature stage. And we looked then at day 66 and day 100 for changes in morphology, so size or abnormalities. And we looked at marker expression for neurons and etc. via RT-PCR and immunofluorescence stainings. And of course, we also looked at apoptosis necrosis. And this is work that was mainly done by Celine Schierke, who was a PhD student in our group, and Kim Jung, who did her bachelor thesis in our group. And this is how it looks like. This is the setting in Cave A at GSI. And here you see the bottles with the organoids. And down here you see a bigger picture. And if you look carefully, then you see the organoids as little tiny balls, bubbles in these flasks. What we observed immediately, well, meaning at day 100, was something that we never really saw in normal brain organoid development and culture. And this is this kind of blabbing, so the formation of cavities. We saw them in all radiation qualities. And here you can see the example for the proton irradiation at day 80. It kind of correlated and with increase in apoptosis and necrosis still at day 100. And so we're trying to see how the effect, how serious the effect was. And so we tried to calculate RBEs. And bear with me, these are preliminary data. Of course, we have to get many more. But what you can see already is that depending on what we irradiate, we get very different RBEs. So what is marked here in red is the carbon irradiation of day 20 organoids. And with the extremely high RBE, which is simply a measure for that the organoids very nicely mimic what is happening in an embryo that is extremely sensitive to irradiation. With the proton irradiation, we looked at two stages. One was irradiation in the plateau phase to have a normal tissue irradiation, and of course the SOBP. And as you can see here, the plateau is basically similar to the X-rays, but irradiating with protons and carbon ions in the SOBP increases the RBE considerably. So considerably more than this 1.1 that we usually get from survival curves. And this irradiation with protons and carbons and the increased RBE, of course, could be linked to radiation necrosis. And we have some ideas how this is actually going on. But this is actually ongoing research. And I hope that maybe in the next talk, someone I can tell you more about this. What we also see after irradiation is a growth retardation. So the organoids are simply smaller in size after irradiation. And this is these are pictures of the proton irradiation. Here you can see this cavity formation from one of these organoids. And this size reduction that is significant for X-rays. For the protons, we need more data still, is linked actually to the loss of progenitors. So we see a downregulation of PAX6, and you can see that here also in the pictures. These are slices of organoids from the control, and PAX6 is stained in purple, and you have the 15 gray sample here. The blue one is the nuclei staining with DAPI. And the same is true for beta-tubulin, which is a marker for immature neurons. And this is also significant. So there is significant change in morphology of the organoids. And if I may summarize this first part about the normal tissue protection, then I would say that the brain organoids are an ideal tool for experiments in radiation biology because they mimic the human brain development so nicely. And the irradiation of brain organoids with different radiation qualities leads to the impaired growth and neurogenesis, but increased morphological abnormalities in the cavities that we see. The irradiation with protons and carbon ions leads to the increased RBE for cavity formation. And as I said, the link to radiation necrosis is work in progress. That brings me to the second issue that we want to tackle, and this is the improvement of tumor control. And here we are interested in the glioblastomas, because the glioblastomas, they are very aggressive, very heterogeneous, very irregularly shaped, which makes them really difficult to treat, and they are strongly invasive in normal tissue. All cells such as neurons, astrocytes, and oligodendrocytes are involved more or less. And despite really the best treatment, meaning surgical resection, chemotherapy plus radiation, the median survival is only 15 months. And I always find it devastating to see the pictures of the brain from these patients. So these huge tumors, and even after this resection and treatment with all kinds of stuff, they die very soon. So that really needs improvement on the tumor control. Unfortunately, proton therapy does not really help so much. In a study of Said et al., the proton beam therapy re-irradiation following surgery and conventional chemotherapy did not really improve much, because the median overall survival still was only 14.2 months. So yes, the proton therapy has all the advantages, as better cell killing, maybe better area control, better normal tissue sparing. But in the end, if the life expectancy of the patient is not really improved, from the patient's view, it is really not what we want to say. And unfortunately, the same is true for carbon ion therapy. Here as well, if you combine photon therapy with chemotherapy and with a carbon ion boost, you get a median survival of 17 months. And the same here. Here is the median survival is 18 months. So this is abysmal. Unfortunately for glioblastoma research, we have, very little in vitro assays that are maybe meaningful. Because of course, what you can do is you can take the cells from the resected tumors and keep them in culture. Unfortunately, they very quickly will lose their characteristics. So this is actually not a good model. Of course, what you can do is you can put these cells, these primary cells of a patient, into a cerebral organoid and hope that the microenvironment of the organoid will keep the cells in a more characteristic way. And yes, that is partially true, because in these settings we have a widespread invasion, and we really have something that looks like a glioblastoma. But you have to keep in mind that you have two very different entities. You have a stem cell, a glioma cell, with a totally different genetic background than the organoids. And you have two different maturation stages. So that's not the best thing that you can do. More physiological would be to induce the tumor within the organoid microenvironment. And to do so, something that is very simple maybe, or simplified, is to induce c-Myc, because c-Myc is overexpressed in glioblastomas. c-Myc is a transcription factor, and it regulates more than 15% of all genes. It's a proto-oncogene, and you can see why, because it induces cell cycle, proliferation, stem cell properties, and decreases differentiation. It leads to high tumor heterogeneity and high grade in gliomas. And so this is something that really makes out the aggressiveness of glioblastomas. And so Esther Schickel, a PhD student in my group, and Tamara Bender, a postdoc, tried to induce brain tumors by c-Myc overexpression. And they used a system, gene editing by sleeping beauty transposons. And what this is you simply have your organoids, and you try to insert the gene of interest, in this case c-Myc, into the nucleus of cells. And you do this with two plasmids. One carries the gene of interest, in our case c-Myc with green fluorescent protein tags. And on the other plasmid, you have a transposase. And these two plasmids you have to get into the nucleus by nucleofection. Then the transposase will cut out the region of the gene of interest and will insert it into the host genome. And what you get then is a green fluorescent c-Myc overexpressing part of the organoid. And if you grow them a little longer, then you end up with these, we call them tumor organoids. But as you can see here, they are very heterogeneous. So sometimes it's just a very faint area of c-Myc GFP overexpressing cells here in green. You have sometimes only one spot, you have multiple spots, and sometimes these tumors infiltrate the entire organoid. And also over time, actually, they are highly invasive, and it's difficult actually to retain a normal organoid part, because of course what we want to have is an tumor in a normal tissue, but we don't want a tumor overgrowing the entire organoid. And so we refined the system a little bit by this scheme. So we induce the tumor, then we select the green fluorescent tumor cells and inject them and transplant them into normal sister organoids. And by that, we can improve a little bit tumor induction and control it a little bit better. And this is what's shown here. So the organoids survive this transplantation perfectly well. And 1 day after injection, you see already a small patch of cells that start growing. And then 8 days after injection, you have a real tumor spot. And this, I think, is a very nice model to actually look at A, tumor control, B, the interaction between tumor cells and the normal tissue, and especially, you know, this is a good measure to look at what's going on in the margin that we always use for the tumor treatment. And first results on these tumor cells show that the c-MYC overexpressing cells retain stem cell characteristics. So here you can see in green the GFP positive tumor cells. Here is also a GFP area here as well. They are counterstained with Höchst as a nuclei marker. But what you can see here in the magnification of this area, you do see that these cells co-express PAX6, the progenitor cell marker, but they do not infiltrate this proliferation zone yet. And they also do not differentiate into MAP2 positive mature neurons. So you see clearly here that we have MAP2 positive cells, but they are not counterstained with c-MYC GFP. And that makes sense. So because these cells really retain stem cell characteristics. And there's, of course, lots of work still to do. But I hope that in a nutshell, I could convince you that organ-like structures of the brain organoids offer state-of-the-art experimental approaches for radiation biology and protection. They can be used to tackle normal tissue control issues. We see growth retardation upon irradiation. We see the depletion of progenitors, and we see abnormalities that can be used and maybe can be linked to radiation necrosis. The tumor induction in organoids is a really nice feature. We see highly invasive stem cell-like cells that mimic glioblastoma features, and we can control this and make it more reproducible. And here the irradiation experiments are ongoing. But coming back to my slide that I used at the introduction, I think that in addition to epidemiological studies and behavioral testing, the brain organoids will really tell us a lot about the onset and the mechanisms that underlie those features that we see, such as radiation necrosis, the detrimental effects on neurogenesis and on behavior. And that by using them in a high throughput manner, it will be possible to find countermeasures that are applicable in the clinics. And with this, I am ready with my, or finished with my scientific part. I thank you already for your attention. Of course, it's great to have really good funders, but it's even better to have a nice research community. And I can say that as a stem cell researcher here at GSI, I really benefit from the interdisciplinary group and department of the Biophysics Department. It's gorgeous to work in this environment. It's, of course, all the work that could not be done without collaboration partners, and Marco already mentioned some of them. I would like to mention the people at HIT because they were very helpful for radiation experiments, especially Stefan Brons. And working in such a great environment is something that, thanks to HITRI, you can do as well. So through the transnational access, you can come and join us here at GSI. I was told that there are contact persons for all of the facilities. Here at GSI, it is Marco Durante and his deputy Konrad Lehmann. And as Marco already referred to, there are research and clinical panels so you can apply for research access and clinical experiments. And at GSI, it is quite simple, I would say. If you want to join us and use our facility and our expertise, you can go to these websites here and apply for beam access to GSI. You have to register with the Welcome Office. You have to file forms and receipts for reimbursement and so on. But you will be guided through all of this on our website. And yeah, I hope to see you here, because I can only say that it is a very fruitful thing to work together with lots of different people. Thank you so much.
Insa, thank you very much. Thank you also for advertising the translational access that, of course, applies to everybody in the HITRI. I think your presentation was maybe a little bit difficult for the physicist, but still conveyed the main message. I think that you can use now a new model system, organoids, to study things that are extremely hot in particle therapy, like radionecrosis, which, as you know, is one of the main issues, especially in proton therapy for toxicity, and of course, the treatment of glioblastoma. So now I'd be happy to have any questions. I already see Manjit.
So Marco, no, so before questions, if you don't mind, can people turn on their cameras so we can have a Zoom photo, please? Thank you again, Insa. Excellent talk. So could we just have 1 minute to do the photos? Please turn on your cameras.
You want to do it now?
But yes, because otherwise people disappear, and I would like to see them.
You're right.
Before they disappear. I presume, Petya, you're going to take the photo, yes, right?
Yeah.
So, Petya, tell us to smile.
Cheese, everybody. Great. Okay, that's done.
Thank you. So Marco, please go ahead. Thanks, Petya.
Okay, so you have no questions, Manjit?
No, I let others ask questions first.
Okay, so we have Michael Kremer. Michael, please.
I actually have two questions, but maybe I can put one after the other. The first one is, you showed a picture where you showed the dependency on ADT of various clinical results, and the gist of it was that you cannot predict anything there, because, or not very much, because the variation between the patients is larger than the effect. I wonder how can you help with organoids there? Do you want to prepare an organoid for all of these different patients, or how should that help there?
Actually, yes. You simply can do a high throughput, which means you can do organoids from iPS cells, many organoids from the same patient, and lots of organoids from various patients. And this is actually something, I mean, it is, you will never only rely on these organoids, but you can do a first preclinical streamlining, and you can see maybe first hints that then you can go back to the clinical studies and see is this what we can see in the clinical studies as well. So
you're saying your procedure is fast enough that you can the patient don't have to wait for weeks until they get treated. So it should be fast.
No, I mean, this is actually something which stem cell researchers try to tackle in that way, that they do biobanking of iPS cells from patients with certain diseases, and that you simply use these biobanked iPS cells already for organoids, etc. So no, you will not have a patient and take his or her cells, make iPS cells out of it and make organoids out of it, because then you will have 2 years and a glioblastoma patient is dead.
Yeah, you don't have the time.
You don't have the time. But on the other hand, still, I mean, from having iPS cells from many different genetic backgrounds, and in this more or less reproducible model of organoids that is not quite as heterogeneous as patients or as animal studies, but at least complex enough, you can draw conclusions.
Okay, so it's not quite personalized medicine yet, but
not yet. No, it will never be with brain
yes.
organoids, because the brain organoids will take 80 days to develop.
Okay, I see. But okay, this is the first step. And the other question was, from your picture, I got it that your organoids are of the size of a petri dish, maybe, or a usual culture flask. Is that right?
No, actually, these organoids are up to 8 millimeters in diameter.
Okay, I see. Very small. Okay.
So they are for stem cell biologists or for biologists, they are huge.
Yeah, okay,
so, but for of course people used to a human brain, they are really tiny.
I don't looking for a complete brain. No, I'm asking because you mentioned this DUD, this uniform dose analysis in the other picture you showed there. And we have just put in the ability to provide this DUD-based optimization into our treatment planning. And of course, this would be a way to verify that if their organoids can be large enough to, for instance, mimic at least a small organoid whisk, so we can maybe measure a difference there. So 8 millimeter is not, well, it's not very large, but it's not nothing. So maybe we can, in the future, we can see something there. Yeah,
I don't know. I mean,
difficult. Okay.
No,
I would try, but I don't. I'm not sure.
I've always been told that measuring cells is old-fashioned. I should stop it. Some people tell me this all the day, and this would be a way to get further in this kind of validation business. So this is why I'm asking.
Yes.
Sure. No. This is definitely. It's definitely a quantum jump compared to cells, but under some aspects, it's even better than animal models, as I said.
Sure.
Okay. Any more questions for Insa? No questions? I hope you got the main point. So Insa, thank you again. Of course, I think that the results on the brain necrosis are already mature for not only for publication, but also for, I believe, changing the clinical practice, because I think it's pretty clear that we have to use at least a slightly higher RB for protons already if we want to avoid brain necrosis. And of course, we all hope that this glioblastoma model will work, because then we can really make a lot of tests for this very lethal tumor. Then if there are no more questions, I just pass to Manjit. Do we know what is the next HITRI seminar?
Not today, looking
today.
for interesting topics. So I'll give some food for thought for the next week.
Okay, okay.
Thank you, Insa. But I wanted to say that I didn't ask a question, but this field that you're just opening up to be able to answer some of the challenges we have in particle therapy and not being able to address those using standard methodology, I think this is wonderful. Really is a great way to try to start addressing some of these. So I thank you very much for your talk.
Thank you.
All right, so with this, we can essentially close. Just a reminder also, we have on Thursday afternoon, so also tomorrow, we have the biophysics seminars on Zoom at 2 p.m. every Thursday. They are also open, so if someone is interested, they can go to our webpage and check. Tomorrow, we will have an interesting seminar from the Danish Proton Therapy Center about flesh. They have very nice results in an animal model about flesh with protons, and the seminar will be tomorrow. If you go to our webpage, you will find all the information. So thank you very much, Manjit. Thank you very much, Sandro. I see also Sandro Rossi here, the PI of HITRI, and I hope to see you again to the next HITRI seminar in about 1 month.
It's about every month, yeah. And actually, and we get a very good attendance, so obviously people are appreciating this.
Yes.
So thanks a lot, Insa.
Thank you.
Bye, bye, bye.
Bye-bye.
Bye-bye.
Bye.
Bye. Thank you.